So, good afternoon, everyone. Thank you for being here. My name is Mariana Perez Bobadilla, but I'm also known as Maro Pebo. I'm an artist, an art historian, and I'm delighted to talk to you about Cosmologies of Care and the relationship that we establish with the others. And during this week, we have been speaking a lot about symbiosis in metaphorical ways. But today I would like to speak about symbiosis in a very literal way, in a biological way, let's say. And particularly within all the forms of symbiosis, I want to talk about endosymbiosis. So what happens when one organism is within another organism? And how does we transform? How two completely different organisms become a third one? How do we transform and acquire superpowers to survive? Through these endosymbiotic relationships. And I would love to tell you that endosymbiotic relationships would allow us to survive, for example, to the climate crisis. But what I'm going to do today is present the theory and artworks that just feature this idea, that evolution is not always about competition, but evolution is many times about collaboration and also transforming, becoming, being flexible into becoming a third one with the other, and about the relationships that we establish with the others that are not always mutualistic, sweet, and beautiful, but are sometimes and oftentimes very painful. So I'm working with bioart or art and biology for a long time. I realize we oftentimes use the term collaborating. So I'm collaborating with the bacteria, I'm collaborating with the plants I'm working with, and we are in our panel very critical to this notion. Are we really collaborating? And what we're questioning is notions of consent and intent. Is the other, is the non-human other really collaborating with us? Is this really a mutualistic symbiosis that we are establishing? Well, most of the times it's not. And what I want to talk about is, of course, symbiosis is about this art of living together. But for all organisms, we are constantly and always living together, as I was saying, not always in pleasant or easy ways. So as I was saying, the term collaboration is used as a comparison to symbiosis when it is mutualistic. But symbiosis also refers to other kinds of relationships like parasitism and commensalism, which is eating each other. And so I think these are better metaphors of what we're doing in the art in the so-called bioart is closer to parasitism and commensalism. So as I was saying, there are many kinds of symbiotic relationships, and not all of them are so pleasant, you know. So we have, of course, the beautiful mutualism in which we all get something from being together, but of course, we also have a lot of competition, predation happening. And for this, I brought one of my favorite examples, the scabies mite that lives inside of our skin. And this is how they look, talking about like extraterrestrial forms of imagining. This happens in our bodies, constantly in human bodies and other species' bodies, and this is how these mites look like. And they dig inside of our skin, and somehow they're forming these kinds of endosymbiosis. So there's many forms of endosymbiosis. Some are permanent. This one is fortunately temporary, so they live inside our bodies. Some endosymbiosis are more permanent or deeper because the creatures come inside our cells. In this case, they're not inside of cells, they're just inside of our skin. But this is a beautiful parasitic symbiosis. So when I'm thinking about symbiosis, I would like to make this turn, not to think about fake collaborations, but thinking about care. And particularly care as a feminist term, but of course within this post-anthropocentric loop that we are all familiar with, which thinks about care about other species, but also the care that is established between species without us. So the works of my colleagues also think about the relationship between microorganisms and machines, and between different species, like what I'm going to show you today. Not always what does this other species has to do with me, but how do other species relate to each other. Most of you are very familiar with the post-human term, but if there was anyone here who's not, the way that I work with it is through the work of Rosie Braidotti, and she thinks about confronting the thinkability of a life that doesn't have me or other humans as a center as an instructive process, right? So it's a shift for an ethics of sustainability. This is the greatest value of the post-anthropocentric term for me. So we can start thinking about the center of my current research, which is this trans-species becoming. how the relating to the others and having them inside of our cells has allowed us to transform. And earlier I spoke about how a viral infection allowed us to develop the placenta and become mammalian. So we have viral DNA inside of us, and that allowed us to transform. This is a very clear of transpecies becoming. But there's other ways of transpecies becoming, and as I was telling you today, the kind of symbiosis I'm interested in is this endosymbiosis, when other organism enters into us to establish these transpecies relations. And I want to speak about a very popular, beloved theme in the present, which is lichen. This is how gorgeous, beautiful lichen looks like, and is one of my favorites at the moment. And as you probably know, a lichen, we consider it one single organism. We even have species name for them, but it's actually at least 3 organisms together. So we have a fungi, we have a photosynthetic partner, which is oftentimes an algae or a cyanobacteria, and we have other organisms all living together as one species. And as I said, we even have scientific names for them as though they were one. But we have all these different, completely different realms of life happening as one. So in this picture, you can see it really beautifully. So we have like the scaffold, which is fungal, and then we have this algal zone. So the fungi gives it structure, and then the photosynthetic element nourishes the whole organism, and this is how it looks in a crosscut. But not only that, microorganisms, lichen are also amazing for astrobiology because they're models for climate resilience if they can resist to space conditions. And as we continue to completely destroy this planet, these extreme conditions of space might be similar here. And as I was saying, these endosymbiotic relationships maybe give us an alternative of how to live, transforming with the others. This is a work that I've developed this year, which is called Lichen Timescapes, which is a series of works that is built by me hiking through different places, collecting lichenous rocks. So some lichen get encrusted into rocks, and I'm thinking about different timescapes. So what we have and what I'm researching is the kind of rock, for example. This one is 600,000 years old. So we were talking about the late Pleistocene. And then we have a species of lichen that is living there. And then this comes from a volcano in Mexico, that is the Iztaccíhuatl. This is how I presented the work. So through collecting the different rocks and through thinking about the times of the lichen, the times of the rocks, and then the expressivity of the sculpture, which is my human timescape. So what I'm thinking here about is human time within biological and geological scale. If I think that a lichen grows for 2 millimeters a year, I'm looking at hundreds of years going on in front of me, which looks like nothing when I compare it to the time of the rocks. So I'm thinking about these timescales and about these very deep relationships that the lichen established with the rock and among the species that live within the lichen. And the real reason that I'm sculpting them is for people to actually look into the lichen, to look into their variety, to have a reason to bring them into the exhibition space and to think about these relationships. So here again, we have an example of this limestone, who is 6 million years old, which is such a depth of time that we can barely conceptualize or imagine, right? And we have a beautiful lichen that I really love, which is called Rhizocarpum geographicum, because it forms these kind of maps on the surface. This is how these little maps are formed, and it's very sensitive to pollution, and it grows really, really slowly. So we can think about the timescapes of the lichen as well. The second project that I'm going to present to you, and with this I'm going to finish, is about bacteria and plants. As you can see, the constant in my work is thinking about the relationships that are established with microorganisms. So with this second work, I found these like small tumors on the roots, and I thought we usually think of tumors as something negative, something that is like destroying an organism, right? But actually what is happening here is a very intense endosymbiotic relationship. Bacteria are coming into the roots, into the cells of the plant, and then they're making the roots go bigger. They become really fat as these tumors. But what happens is this gives a superpower to this plant of bringing in, as you can see, the cells are all like swollen, and they're capable of fixating nitrogen from the air into the soil. So the plants that develop this relationship with the bacteria are way stronger. So I wanted to bring this to the world because this only happens under the ground, and it has political dimensions, because when we have monocultures and we don't want to have legumes, but we only have fed for animals, we are like skipping on these relationships. So I wanted to show this that is happening underground, so I developed this series of sculptures. As you can see, my lichenous rocks are still there from my collection, and I developed these sculptures in which the glass melts into the rock to think about the relationships and how uncomfortable are the relationships, how much do we have to bend and transform in order to adapt to the other. But at the same time, we could see what's happening in the roots, to reappreciate something that is hidden, that's something that looks trivial, but is this becoming in the other, allowing the bacteria to come into the cells of the plant and making it stronger, or giving it the superpower of fixating, in this case, of fixating nitrogen. So this is how the series look like. It has all the challenges of living art, of keeping the plants alive. Originally, I wanted to display the roots and the nodules outside, but when you grow the plants after a while, you start feeling a relationship to them and you no longer want to kill them. So this is what happened. I end up devising a way of keeping them alive. And this is how the work looks like. So in order to, as a conclusion and giving the space for my colleagues, I wanted to present this idea of symbiosis, not only as a pleasant, mutualistic thing, but also as a way to transform ourselves. So how endosymbiosis allows us to transform and establish trans-species relationships. Thank you.
There you go. Wow, hey, hi everybody. Thanks for coming today. These are really short little talks, and we were concerned that we didn't have a lot of time to discuss at the end of the talk. So we're actually inviting all of you guys that might want to talk about what we talk about to meet us for a drink at the end of the symposium today. We're gathering at Cafe Flore, which is right next to, if you're facing Les Halles, it's directly to your right. It's the bar covered with all the fake red flowers, so if you have any comments, come and join us. My name's Elizabeth Demaray. I'm an artist, I'm a professor of intermedia, I knit sweaters for plants. I'm working on making emergent ecosystems on the tops of modernist buildings in lower Manhattan for our non-human companion species. I code, I make listening stations for birds that play human music so that songbirds can use Pandora radio, and I culture lichen on the sides of skyscrapers in New York City. So I'm going to very quickly talk about a couple of pieces, but most of these works involve the concept of a biotope, which is a small environment shared by multiple species, often including humans. The works also involve the idea of trans-species giving, which is this idea that asserts that the commonalities that exist between living organisms are such that we might be able to be of assistance to another life form. And also these works may involve non-anthropocentric design, which is design that attempts to consider the aims, desires, and perceptual landscapes of the non-human. So in this, in the next few minutes, we have a couple of questions, and I'd actually like to get your guys' thoughts on this. We've been thinking a lot about this. I've been thinking a lot about this. The first one is collaboration. Are we really collaborating with these other life forms? The second question is learning outside of a scientific model. You know, what are we learning here that's outside of, you know, a model that generates factual information? And lastly, this comes from Robin Wall Kimmerer, and the question is, can we learn from nature rather than about nature? So, you know, once upon a time I was a regular old everyday sculptor. I made objects because they communicated things, and I had this really fateful day. This was 2 decades ago. I was teaching a conceptual art making class at the University of California at Berkeley, and I told my students to bring a really important object with them to class. And one student brought a beautiful shell that she had found on a beach in Santa Monica, and I joked with her that some hermit crab was going homeless because she brought this shell in. And another student, who was a microbiology major named Jose Dennehy, brought in a really spindly, sad-looking little plant. And he excitedly told all of us that he had genetically altered the broccoli plant. So you're seeing a picture of a nice healthy broccoli plant. This one was really spindly, and he told us that he had disconnected the plant's ability to reproduce, and he had given it a pre-programmed death cycle of 6 weeks. And I was just overwhelmed. I had already been knitting sweaters for plants. I love potted plants. I love the idea that as humans dig something up from outside, we bring it into a domestic space, we care for it. And I was kind of overwhelmed. He gave me the organism. Apparently he wasn't supposed to take it out of the laboratory, but he gave me the organism. I took it back to my sculpture studio. I left it on a bench, and I watched it slowly expire over the course of about 2 weeks. And this got me thinking that maybe I could use my skills as a sculptor to potentially help some other kind of life form. So I started doing a little research, and it turns out that hermit crabs actually were experiencing a huge housing shortage. This was 2 decades ago. Right now it's much, much worse. But this animal has its growth spurt in the springtime, and at that time I think it's 50% of all hermit crabs are living in shells that are too small for them. A lot of this is due to human degradation, environmental issues. But they find them living in all kinds of forms of refuse in order to remain housed and protected from predators. The interesting thing about hermit crabs, though, is because they exhibit choosing type behavior, scientists actually knew exactly what they liked. And what they like are forms that have a lot of internal space to weight ratio. So I took the earliest form of AutoCAD and I designed what I said were better designed forms for this life form than what nature had ever presented. And we made them with some early form of stereolithography. It was a big success. We showed it in a museum. 25% of the crabs that were in the population chose a man-made home before the end of the exhibition. And the interesting thing, the art part about this is really a design question, but the art part about this is that, especially back then, these were very expensive forms to create. And in attempting to figure out how to pay for it, we proposed putting a corporate logo on the front of each man-made house before placing it back into nature for the animal to use. And the idea was that, struggling life forms that aren't very important to us humans might need to actually apply for corporate largesse in order to continue. But I've been thinking a lot about this project lately because it was the first and actually the last time that I ever had another life form that exhibited choosing type behavior into my art studio. And the interesting thing was I actually learned a lot about the animal, but I learned the kinds of things that you don't read in scientific literature. It was sort of like having a bunch of really difficult house guests. There were certain entities that would only eat a certain kind of papaya. There was another one that would only eat chopped up frozen goldfish. They had lots of different kinds of behaviors. I had to modify everything I was doing. It was really exhausting. But that was the first and the last time that I actually had one of these life forms into my studio. That's calcium carbonate, which is the sort of the grail in terms of creating these sorts of forms. So the piece was called the Hand Up Project, attempting to meet the new needs of natural life forms. The next work I'm going to talk about very quickly is 4 potted plants. And it's sort of an homage to Valentino Brattenberg, who was an Australian-Italian cyberneticist who, through a series of thought experiments, created these very simple vehicles for avoidance and attraction. He showed that these very simple vehicles actually seemed to perform behaviors that looked a lot more complex. In any event, myself and a team of scientists created these things called floorboards. This was back in 2011. And the entities are part plant and they're part robot. You can program into the robot the needs of the plant that it supports. It's a learning system. It learns where sunlight and water is in the immediate environment. These navigate around in the engineering department. They get under people's feet. They're difficult to get around. You can interact with them. I'm really interested in potentially making these, supporting these with new forms of AI. Back then we were just using computer vision systems, and we were figuring out how to make self-watering plants, and it turned into a lab which is called the Art and Artificial Intelligence Laboratory at Rutgers, which is a platform for the use of AI and computer vision in art. But one of the things that I thought was really interesting is we were attempting to get lots of funding to make more iterations. And the interesting thing about plants is that they're really sensitive to environmental perturbations because they're stuck in one spot, usually. And they're really good at gas exchange, they're really sensitive. So we started looking at electrical potentials in the face of different kinds of environmental perturbations. And what we found, without a lot of long-term sort of sedimentary gathering of information, what we found was that the plants actually looked like individuals. They all sort of individually had different signaling, different kinds of signaling based on the kinds of environmental perturbations they were experiencing. So I thought that was really interesting, that we couldn't make generalities, that where we were at was a situation where we were actually getting to know sort of these individual entities. So the last piece I'm going to talk about quickly is something that I'm doing right now. I'm pairing with a scientist, her name is Dr. Amy Savage, and this is for urban living pavement ants. So they're the kinds of ants that would be living on a median like this in the middle of a city. And the interesting thing about these ants is that they're very hostile, they're angry, and they don't get enough protein and fat in their diets because they're living off of the kinds of food that us humans drop. And they're really important. They do all kinds of important ecosystem services for us. But because they're living on what's called bonanza food supplies, that's when we drop some pizza crusts, soda pop, lots of sugar, lots of refined carbohydrates, they're very angry and hostile. And they will basically attack and eat all the other environmentally supportive insects in the immediate environment, except for a couple species of beetles that have really hard exoskeletons. So in any event, we're creating these ant feeding stations that allow local communities to adopt colonies of urban living pavement ants. And at these specific sites, you can leave small amounts of healthy protein and fat for the animal. And my students are creating an app called Antsy, sort of like Etsy, but only for ants, where the communities can log in the food sources. I'm interested in creating an architecture. Apparently no one has ever made an architecture to be shared by ants and humans. But I've been thinking a lot about Louis Kahn, who sort of was interested in allowing us to experience the space almost as though we were ants in these large spaces. So in any event, we're proposing to put this at the Salk Institute, which has these beautiful tiles that have tunnels underneath them, which sort of allows us to interact with the species at the same time. And that's the end. Oh, we're hoping to learn from nature rather than about it with this piece. So that's another one of our aims. And also, how do these animals actually live in the built environment? And is it possible for us humans to alter their lives to support them in such a way that they can flourish or potentially become an overpopulation? We're not sure, but we're looking forward to seeing what we find out. So thank you all.
Oh, okay, there it is. Okay. Thank you. So my name is Carlos Castellanos. I'm faculty at Rochester Institute of Technology. I'm an artist. I work with technology, with AI, and more recently with quote-unquote primitive organisms like bacteria and plants, which I'll talk about today. So I'm gonna just talk about a couple projects and how they're sort of like little models for what I'm calling an ecological AI. So just for a little context, I've been inspired a lot by the work of a cyberneticist called Gordon Pass. Maybe some of you have heard of him. So he and I'll show a brief sort of example of it. He created these electrochemical systems that were basically kind of like computers, what we will today call computers, and they adapted to their environment and they learned. And he sort of kind of proposed, or had ideas of kind of like a different kind of science, a sort of, it's just second order cybernetics, a kind of accounting for a subjectivity in science. And he was interested, as were other cyberneticist colleagues at his time, in kind of sort of adaptive, what I'm calling adaptive sort of conversations with the environment. And the term conversation here means, I don't use the term collaboration, typically. Conversation in this sense means action-grounded construction of shared meaning. So like the knowledge isn't just out there, it's constructed. And Andrew Pickering calls it a sort of a basis for a new art science. So, so he, like I said, created these electrochemical systems. His colleague, Stafford Beer, who he worked with, again, maybe some of you have heard of him, he experimented with daphnia and leeches and sort of, and kind of enlisting them to become sort of interested in us in some way. And he experimented with what you might call creating pond brains. So again, same idea, kind of adaptive conversation with the environment, and in his case, again, primitive living organisms. And I actually created a project that was a kind of recontextualization of past electrochemical systems where we, set this up in a gallery and fed it environmental input, sound, motion, etc. This is at SIGGRAPH in 2012. That's a close-up shot of the electrochemical growth. Basically, you put voltage, current in, and it's sort of like growing wires. This is stannous chloride. So, and there's a lot of kind of, again, primitive organisms that that we can choose from, right? And but again, this is just for context. So this is like a complex adaptive systems view of life and intelligence. So this is not, so cybernetics and AI are typically sort of historically they're thought of as sort of coming from the same place, but they really have different kind of epistemological and ontological kind of roots, really. So again, this is for more for context, but this is the kind of the view on a complex adaptive systems view of life, where we have these kind of living system establishing adaptive linkages with their environment and in these embodied flows of embodied networks and flows of information. So that's not really how AI works, right? So the first project I'm really going to talk about is called Beauty. So we were experimenting with these pattern-forming bacteria. I should say at the beginning here, this started life as a residency at the Coalesce Center for Biological Arts in Buffalo, New York, in the U.S. So Beauty is a hybrid machine microbial artwork. It features a bio-driven artificial intelligence that remediates a contaminated soil ecology while and doing so creates a situation where the fates of the contaminated soil and the bacterial cultures are determined by the whims of this AI, a deep reinforcement learning agent with an internal model of beauty. So the agent builds this model by observing the pattern-forming behavior of these bacteria that exhibit this sort of collective social motility and produce these intricate growth patterns. So the agent attempts to spatially modify the bacterial growth by introducing chemical attractants and repellents, right? So the colony growth and movement, like I said, are captured by the agent, and they determine and analyzed to determine how well the colonies conform to its model of beauty. So the more beautiful the growth patterns appear to the agent, the more of a remediating solution the soil receives, and the more nutrients the bacterial cultures receive. So these images, these patterns are basically like they're sort of searching for food. It's a search, essentially, right? So it's known that they only produce these patterns under kind of an environmental stress, sort of lack of food or moisture. So the takeaway here is that in order to, so they have to starve themselves in order to look beautiful for the agent. That's the idea here. Go to the next one. So this is just a system diagram. So we're using, like I said, a deep reinforcement learning agent. I won't go into the details here. We can talk about it later. It's based on what's called the world model. So we basically have 3 machine learning models: an autoencoder, a recurrent neural network, and I guess a vanilla neural network as the controller. And this is this project was part of a group, sort of a experimental research collective called Phylum that I'm a part of. So check out our website. And then next project I want to talk about was part of a residency I did in Spain last year, just finished a few months ago in Valencia at a plant biology research institute at the Polytechnic University of Valencia. So lots of plants, lots of greenhouses, and lots of learning for me on how to basically manipulate plants, injecting them, as I am doing here, with agrobacteria to introduce. It's a bacteria that kind of acts like a virus and introduces, basically, you modify the DNA of the plant. Just the leaf, it's sort of transient. So once the leaf dies, the change goes away. So I created this project called Post-Natural Prototypes. It's what I like to call a plant futures garden. So I was working with scientists, and we genetically modified the plants so that they trigger observable morphological changes as they grow and are subjected to environmental stress, like drought, which is a lot of what they researched there, right? And then we trained a machine learning model. In this case, it was we're doing clustering, unsupervised clustering, to transform that. So they'll recognize these changes and transform them to sound. The idea is that the plants sort of respond sort of like they're crying or something, right? Or talking, right? And also change their environment, like adding water or not adding water, or changing the temperature, adding hormones inducing drought with this chemical called polyethylene glycol, which sequesters the water so the plants can't get to it, the plant roots can't get to it. So again, it was just this sort of back and forth between the AI model that's sort of controlling the plant's environment to a certain extent, and then you're sort of hearing it. So we're translating it in a way that humans can sort of understand what's going on. And it's a bit of a kind of a de-anthropocentric sort of stance in that the plant agency is given a bit of a higher status, and the plant AI interactions are not. I tried as hard as I could to have the model not be sort of optimizing. It's just a conversation, not a collaboration, but a conversation. So there's just some more images of the installation. It's sort of like you walked into a garage 50 years in the future, and these are the only plants left, or something like that. So the 2 images here on the right here are seedlings. So we grew seedlings for 2 of the prototypes, and they're genetically modified by putting in what are called reporter genes or reporter proteins, basically enzymes that are, on the top one is luciferase. So when it's subjected to stress, releases a hormone called ABA, abscisic acid, and it basically lights up. There's no light source there. That's the plant itself lighting up. And it's like a 10-second exposure. So it's not a lot of light, but you can capture it. And on the bottom, it's GFP. And these are Arabidopsis thaliana, a model organism, and it's used a lot in plant biology. So yeah, so that's the luciferase, and this is the GFP. And we also did one with thermal sensing of plant stomata, so with thermal cameras. And when they're subjected to stress, they tend to close their stomata, so the leaf temperature will go up like 1 or 2 degrees Celsius, so we monitor that. Oh, and then here's some sounds. So every once in a while they would do that. One more. Okay. So this was a team effort. Like I said, I collaborated with scientists and some students helped me out. So this was exhibited at the Centro Carmen de Cultura Contemporánea in Valencia. So these are just little sort of models of what, again, I'm calling an ecological AI. You know, can we have kind of these open-ended flows of information between this sort of primitive organisms and AI systems, right? That are not, they're not optimizing. They're just, again, having these open-ended flows. And I'll just end with a quote that sort of, I guess, inspires me, that what we're trying to, I think what we're all doing here is trying to construct canons in contemporary art, this is Andrew Pickering, that draws on science and technology as its substrate, but in its products radically subverts the ontological premises of modern science and engineering. Thank you. Oh, yes, questions? I guess there's no moderator. So. We self-moderate. Yeah.
Oh, I think somebody will come and bring you a microphone if you have a question. Yeah. But we can probably hear you. You can probably just say it.
Yes?
Thank you. Wonderful to hear about all the projects you're involved with. That's very exciting. My most pressing question was, what time are we meeting for drinks?
Oh, yeah.
5 o'clock.
Well, yeah,
I think 5 o'clock.
Yeah, Cafe
the...
Flore, F-L-O-R-E, that one?
It's called Florida.
Florida. Okay,
Florida.
Good. I'm glad I asked.
When you walk, when you're facing Les Halles, and there's the big churches to your right. Yep. It's just slightly behind the big church, covered in fake red flowers.
Fabulous. I particularly enjoyed the plant robots. Reminded me of a project that was done with a goldfish, and the goldfish would drive itself around.
Ken Ronaldo. Yeah,
yes.
Really fabulous. Yeah.
So I'm just curious about the next iteration of that project for you. You said you were building some AI systems around that?
That project took even simple. I mean, that was 10 years ago now. It took a lot of money. NASA paid for those. I actually had to. I told them in the grant that I was making plants that could grow all by themselves. And that got me funding. So yeah, it's a funding question, but I'm really interested in this idea of what sort of supports would our current forms of AI, what kinds of supports can they actually can they be used for living entities? What kinds of deep knowledges are accessible through those kinds of interfaces? So that's a hope. If anyone wants to fund it, let me know. Come talk. Yeah, do we have questions for each other? I have to say, I love the lichen. Those rocks are just beautiful. Wow, I just love the idea of the sort of the deep time. What made you think about lichen in terms of deep time?
Well, I'm thinking about, of course, we're super technologically mediated, and all the art that I've been studying for a long time is as technologically mediated as possible. And we have these incredibly complex mechanisms and bioreactors. And I thought, like, how can we speak about these same topics just with the least technology possible? And for me, that was the challenge, to make something that was more formal and that it could exist and talk about what is already very complex biologically without so much mediation. As much as I love, the devices and the magic of it, this was like the challenge that I was addressing in this instance. Yeah, so.
Those are beautiful sculptures. Really
yes,
lovely.
Beatriz. Yeah.
Thanks a lot for the talks. I have a question for you, Elizabeth. I liked a lot this non-human architectures that you're thinking about, also this pavement ants, how to make them a construction for them or a house for them. I was wondering, when I also encounter other forms of non-human architecture or design, that sometimes the artists are facing that actually the construction that I built for the specific species is not being used by this, but by others. And then I was wondering if you also encountered this kind of form, that this ant pavilion was then used by wasps or bees or whatever, or rats even, and attract then other problems, let's say. And so this kind of, we would like to help those ants, but on the other hand, how much we actually help them, and maybe it goes in a completely different direction.
That is such a good question. There are people, there are a couple of really interesting designers that are working right now. There is, where is she? I think University of Buffalo, really interesting architect who's making habitats for bats. You know how like bats like to go live in the rafters? She's created just the roof of a house with nothing else, so that it's sort of applicable to bats. But your question about what else could move in, we haven't made these yet. The interesting thing is the idea of making something that's high enough so that humans can easily access it, but also accessible from the ground for ants. We actually, I didn't go into this, I don't want to do a lot of flat horizontal surfaces. We don't really want the colonies to move in to the feeding stations. But it's going to be really interesting. What else might use those spaces? So it's a great question. Right now, all we've been doing is just feeding them flat on the ground. Difficult for people to find the feeding spots when you do that. So yeah, but it's an interesting question. We'll see. We'll see what uses it. Thank you.
Hi. Thank you very much for your presentations. It was very inspiring. And I was wondering about the topic of cosmologies of care, and I wonder how you feel or what your thoughts are on this relationship between, in one hand, taking care, and in the other hand, being careful of. And I feel like this kind of relates with what Mariana Perez was saying about the different types of symbiosis. And I would like to hear about how you think about that in your work. Thank you.
Thank you very much for the question. For me in particular, it comes from, of course, this feminist thinking. What is the place for care? Like a task that is not generally valued in organization that values profit over everything, and how this work of care has been left behind, and how this participates in the post-human turn. So how do we think about care within, like in trans-species relationships or interspecies relationships? And I think it's beautiful what Elizabeth is doing, right? So how are we thinking about other species and their needs in a very, let's say, basic form of thinking the cosmologies of care, and how can we translate it into the others, and how do the scaffold of the lichen is taking care of their photosynthetic partners, for example, or how the plant gives this chemical signal and the bacteria recognize it and they recognize each other. There's all these micro processes of care that are taking place all the time, besides these processes of symbiosis that can be really cruel, of commensalism. But there's also all this constant process of very subtly perceiving the other, and relating to the needs of the other among species subtly. And that I think is great if we would pay attention to these connections, also for us establishing other trans-species connections and thinking of care in this dimension. But yeah, thank you very much. I don't know if you want to add something in the way that you relate with care, with plants and things.
About care specifically, I mean, the plant, the project I did in Spain with the plants, that was part of a residency. So I was doing all kinds of stuff. What you see is not like, kind of, it's sort of what I could, I guess, get away with with doing, or I felt comfortable enough, sort of exhibiting. I don't know. I mean, I don't know if I have a good answer. All I try to do is give, maybe it's a little cliche, but sort of present a kind of interface window to, for us as humans, so it still might be sort of anthropocentric, but to experience some of this otherness, right, in a, as much as possible, a horizontal way.
I just want to follow up with that. I'm also very interested in the relationship between care and control. When I was designing those hermit crab houses, for example, I was looking at, I think that the modernists owe the fascists a lot. And I was looking at Giuseppe Tarani's work, the Italian fascist architect who did the monument to Mussolini. And so I was really interested in this idea, like the fascists had this notion that, that they knew what was good for everyone. And because they knew what was good, they were going to, sort of impart their ideas on everyone else. And I do feel that that's a model that as humans need to think about. Interestingly enough, I actually had IBM at one point wanted to fund me making hermit crab houses. And then they went and they saw that I was talking about this idea that I was, looking at, this fascist model as care versus control, and I lost my funding. So, don't talk about that. Don't talk about stuff like that. But those are the kinds of things that I think about when I'm doing this kind of work.
I mean, and that's at least a subtext of some of the, the big AI firms like in the US, like OpenAI and all these. There's a subtext there of kind of, there's people, researching this, they want to improve humanity and all this stuff. And it's really a kind of, there's eugenicist roots to it,
And also a lot of the implication is that we understand what the other needs when you're taking care of them. We are imagining the desires of the other, of the lichen, of the plant, of the roots. We're imagining as well as we can the desire of the other, but this is always a risk in this process of care.
Right.
Yes, there's a question over there.
Hello, thank you for the part that I have seen. It's an amazing presentation that I've just been witnessing. I had this question in my brain before I came here, and I want to lay it out for you. Since I love this whole living systems and the nanoelectrical element that was in the last or in the pre-last presentation, I'm always looking for this thing that was in the 80s. It was a film about an elevator in the Netherlands that went wild because they had used nano artificial intelligence, so biological artificial intelligence. And it was thinking it was attacking the passengers of the elevator. Of course, it was really kind of over the top, but did you ever think about this element of artificial intelligence within the biological sphere that would actually learn itself all kind of destructive behaviors, a little bit ant-like?
Well, okay, so a lot of sort of AI takes a lot of, science, computer science in general takes a lot of inspiration from the natural world. I mean, deep neural networks are crudely modeled supposedly on the brain, convolutional neural networks on the mammalian neocortex, things like that. But all that adaptive and emergent stuff goes out the window. They don't care about it, actually. It's like a black box, and as long as it optimizes, gets me around, it's fine, So again, what I was saying before that I was trying to do is, you have to kind of jerry-rig these things to use them in this manner that I'm trying to use it, and it's a lot of trial and error, and you see it in the work itself. It looks like a prototype, and that's the intent. So yeah, so I always think about the biological, I'm going to say distortion of biology. I'm trying to, just imagine a different world like the ones that Gordon Parsons and these others were imagining. We don't live in that world. We don't have pond brains, right? Maybe if we did, I don't know, other things would be better.
I'm not sure if I understand the example you're talking about, but when we're thinking about biocomputing, for example, so we're using like moist-based models for there's two things, because all the definitions of artificial intelligence are talking about imitating human brains, whereas what they're actually doing is are also imitating other forms of intelligence. Going back to the non-human, there's all these hive and other species forms of intelligence that are actually simulated and processed through what we call artificial intelligence. But there's other strength that Carlos might know much better than me, which is this of biocomputing, when we're actually using other organisms as the substrate for computing. And of course, this is bound, and as well as as algorithms that we use to go in directions that are outside of our control. And going back to the question of control, right? How much are we in control, and how much do we allow other species to be in control and to think, and we might not have the results that we originally intended. And the scenario that you're posing is a little bit like terrifying, right?
Science fictional, eh?
Science fictional, almost. But of course, I think it's great. I think I would be open to let control for other species. I don't think it's so bad,
If you really are interested in biocompute and actually literally making research into making computers out of slime mold or mycelium, look at Andrew Adamatsky's work at.
Can you repeat that, because you
Andrew
spoke fast?
Adamatsky.
Andrew
And he works a lot with artists, actually. So he's a crazy guy.
You also asked if we had thought about that particular scenario. And I have to tell you, I've never thought about it, but while you were talking, it gave me the idea for an artwork. So
Oh my
thank you.
goodness.
Are there any other questions? Anybody else? No? Should we conclude? Are we good? Yeah? All right, well, thank you all for coming today. Thank you very much. It was a pleasure. Bye.